Caffeine may not have started its career as an anti-aging molecule, but it's quickly becoming a muse for drug designers looking to bottle the benefits of metabolic stress without the buzz. Its ability to activate YMPK, improve mitochondrial efficiency, reduce inflammation, and delay cellular senescence has put it on the radar of pharmaceutical researchers aiming to slow down aging at its roots. The new frontier isn't just about eliminating wrinkles or extending lifespan for the elite. It's about creating compounds that mimic caffeine's behind-the-scenes molecular strategies to promote healthier, more resilient aging across the board. At the center of this movement is AMPK, the metabolic master switch that caffeine reliably flips on. When AMPK is activated, it tells cells to halt unnecessary growth, conserve energy, burn fat, and clean house. These actions are highly beneficial in aging tissues, where energy becomes inefficiently used, inflammation creeps in, and cellular junk accumulates. Naturally, YMPK has become one of the hottest targets in pharmaceutical aging research, and caffeine's reliable, low-grade activation of this pathway provides a compelling blueprint. Drug developers are exploring direct YMPK activators, compounds designed to simulate the metabolic state induced by fasting or exercise without requiring a lifestyle change. One such example is ACAR, an AMP analog that was one of the first molecules shown to directly stimulate AMPK in muscle cells. Although promising in animals, ACAR's use in humans is limited by short half-life and off-target effects. Now, more refined AMPK agonists are in development, aiming for tissue specificity and minimal side effects, essentially trying to isolate caffeine's benefits while leaving behind its jittery stimulant effects. Another strategy is to target upstream regulators of AMPK. Caffeine doesn't act on AMPK directly. It modulates adenosine signaling and intracellular calcium levels, which influence the AMP/ADP ratio, indirectly triggering AMPK. Some pharmaceutical approaches are now focusing on adenosine receptor antagonists, similar to caffeine but engineered to be more potent and selective. These compounds may offer a more precise way to tap into caffeine's anti-fatigue and metabolic effects without overexciting the nervous system. Closely connected to AMPK is the broader mitochondrial stress response, another aging hotspot. Caffeine helps improve mitochondrial efficiency, partially by supporting mitochondrial biogenesis and reducing oxidative stress. Drug companies are now investigating compounds that mimic this effect more powerfully, such as PPAR coactivators, SIRT1 activators, and urolithin A analogs. These aim to rejuvenate mitochondria in aging cells, reduce reactive oxygen species, and improve cellular energy metabolism, just as caffeine appears to do, albeit modestly. Pharmaceutical mimicry of caffeine also extends into the realm of senescence modulation. Caffeine's potential to delay cellular aging by reducing DNA damage signaling and dampening pro-inflammatory cytokines has inspired interest in drugs that can suppress or reprogram the senescence-associated secretory phenotype, SASP. Emerging xenomorphic drugs aim to reduce the inflammatory footprint of aging cells without killing them outright. If caffeine can do this in part through AMPK, future drugs may amplify the same outcome by targeting additional stress-regulating pathways like p53, NF-B, and mTOR. There's also a metabolic dimension. Caffeine modestly improves glucose uptake and fat oxidation, which protects against type 2 diabetes and metabolic syndrome, two major accelerators of aging. Drugs like metformin, another indirect AMPK activator, have gained traction as anti-aging candidates for this reason. But newer compounds aim to go further, combining caffeine's mild metabolic effects with enhanced insulin signaling and deeper regulation of lipid metabolism. The challenge is creating compounds that offer caffeine's cellular benefits without its system-wide stimulation. Caffeine's widespread presence in the body, including the brain, heart, and adrenal system, makes it both versatile and unpredictable at higher doses. The goal now is to design targeted mimics that work at the tissue level, YMPK activation in liver and muscle, mitochondrial tuning in neurons, anti-inflammatory signaling in skin, and immune cells. Some researchers are even considering delivery innovations, like topical formulations for skin aging or nano-encapsulated AMPK activators for brain aging, essentially engineering caffeine-like benefits into specific aging tissues without overwhelming the rest of the body. In the future, a caffeine mimetic drug might come in multiple forms, each designed to target a different node in the aging process. So while caffeine remains a humble molecule from the plant kingdom, its cellular mechanics have inspired a modern medical strategy. Use smart, subtle stress signals to activate repair, boost resilience, and hold off degeneration. Caffeine doesn't force the body into youth. It nudges it gently, reminding cells to adapt, clean up, and keep functioning. The drugs inspired by it aim to do the same, but with pharmaceutical precision, broader effect sizes, and fewer bathroom breaks.